Good afternoon, good morning, everyone. Welcome to our Sunstar Conversations Pro Series. My name is Rachel, and I will be your host today for this 9th and last webinar of this year. First, if you are joining us through the registration platform Eventbrite, you may want to switch to YouTube by following the link at the bottom of your screen in order to enjoy the full screen mode and also ask your questions in the chat box. Now, over the past months, we have welcomed various experts to discuss oral health challenges at different life stages, such as behavioral change in middle-aged patients, gingival inflammation in pregnant women, oral frailty in aging patients, and so on and so forth. In November, we decided to cap off this series with two specials to tackle what we call the perio link, the interrelationships between oral health and general health. These two specials are done to celebrate the winners of the World Perio Research Awards, which are sponsored by the Sunstar Foundation. A couple of weeks ago, we had our first edition with the winner and the runner-up, who discussed about the link between oral health and cardiovascular diseases. Today, we will celebrate the third winning paper, which is entitled Inflammatory Bacterium and Oral Squamous Cell Carcinoma. And to do so, we are very happy to welcome for the moderation Dr. Nicholas Jakubovic, who is part of the selection committee of the award and also editor-in-chief of the Journal of Dental Research. Hello, Nick. Thank you very much. Thank you very much for being with us today. Nick, as you are part of the selection committee of these awards, would you like maybe to introduce these awards to us?
Certainly, I'm delighted to be here today representing the Journal of Dental Research at these awards. So the award has been running for over 20 years now. It was initially introduced by Dr. Bob Jenko in the year 2000, and the first awards were made in 2003. So we're now on our 7th cycle of the World Perio Research Awards, and these are supported by 4 different journals in the field of periodontology and dentistry. So as you may have seen in the introductory video, the Journal of Periodontology, the Journal of Periodontal Research, the Journal of Clinical Periodontology, and the Journal of Dental Research all support these awards. And I'm particularly delighted to be moderating this session, which is on the oral microbiome and oral squamous cell carcinoma. My own research interest is in oral microbiology and the microbiome and the impact of that on oral and systemic diseases. So I'm looking forward to discussing this topic today.
Yes, indeed. With your specialty in oral microbiology, your input will be quite interesting to have on this paper and to discuss with you on this topic. Who best than the authors of the article? So we are happy to welcome Dr. Manosha Perera, who is the lead author of the paper and who will be joined by one of the co-authors of the article, and who is singularly enough to be mentioned, also her twin sister, Irosha. Welcome to both of you, Manosha, Irosha.
Yes, most welcome. You are most welcome.
We are very happy that you accepted our invitation to this webinar. So please let me first introduce you to our audience. Manosha, you obtained a PhD at the Griffith University in Australia, and you are currently a freelance research scholar specialized in oral carcinogenesis and microbiology. Irosha, you are a dental public health specialist and the head of the preventive oral health unit, at the National Dental Teaching Hospital in Sri Lanka. So again, thank you for being with us. And first, congratulations for winning this third place at this prestigious award. Maybe so as to have an idea, could you set the stage for us and give in one sentence the summary of your findings to our audience? Manosha, maybe.
Yes, I would like to give a one sentence summary. It's just like having the first spoonful of your favorite dessert, let's say. So, in our award-winning study, that study shows dysbiotic inflammatory bacterium associated with the OSCC tissues, different species, but with a same function, that is promoting inflammation by plethora of virulent factors. So that is the one sentence summary of our award-winning research.
Oh, yes. Enough? Thank you for this summary.
More on the way, many explanations are there, right? Just like that's why I said one scoop of your favorite dessert. Yes. Now let's see. How
indeed.
Full dessert. Let's say cup of dessert. Yes.
Indeed. Thank you for this summary in one sentence. But obviously now we would like to know more on the paper. And so let's start the conversation. And just before I let you all three deep dive in the topic, may I just ask you to give us a little bit of background around the study. What brought you and your team to this topic and to this research specifically? And then I will all let you discuss.
Yes, it is a very important question. So the first and foremost, this is one of the specific objectives my PhD research. Then the second reason, advancements in next generation sequencing of omic technologies related bioinformatic analysis of high dimensional data have provided an opportunity to view microbial communities in an unprecedented breadth and depth. So, researchers have started doing microbiome studies, especially in the past within past decade. At the same time, humans are called as superorganism or holobiomes. The reason is the metagenome, that is the genome of all microbials, normal inhabitant. Here I am not mentioning about pathogens. I am not relating to pathogens. These are normal inhabitants of our body in various ecosystems. The gene content surpasses the human gene content by 150 folds. That means metagenome is by gene content much bigger than the whole human genome. Then, with these microbiome research are progressing, it has been found disease-associated specific microbiomes in inflammatory diseases, ranging from inflammatory autoimmune diseases like multiple sclerosis of CNS, then cystic fibrosis of lungs, then interestingly, these inflammatory metabolic diseases like type 1, type 2 diabetes, then obesity. At the same time, few cancer types, that is oral, colon, pancreatic, and gastric. Not only that, even in breast cancers. So these research findings tempted me to do my PhD study based on this microbiome. At the same time, when we come to epidemiology, yes, Dr. Irosha she is the ideal person to give about the details about the status, incidence, prevalence of oral cancer in Sri Lanka. This is very important for all the clinicians all over the world, I suppose. Dr. Irosha, please explain.
Yes, the other most important reason why that Manosha based her PhD on these oral squamous cell carcinomas in Sri Lanka. Sri Lanka is a lower middle income developing country, and oral cancer is the number one cancer among males in Sri Lanka, and number 8 cancer among females in Sri Lanka. Actually, it is associated with well-known lifestyle-related risk factors such as betel chewing with or without tobacco, smoking, alcohol consumption, and despite the emerging importance of this connection between poor oral hygiene and the periodontal disease burden and its link with the burden of oral cancer, being the number one cancer among males in Sri Lanka, still there was no research that connected the periodontitis or the periodontal disease burden or the oral hygiene status, which could be an important emerging risk factor in the existing risk factor profile of oral cancer patients in Sri Lanka, because now all these lifestyle related risk habits are modifiable, and in the same vein, if periodontitis and poor oral hygiene is if it emerges as an independent risk factor, still that also can be modified by improvements in oral hygiene and the periodontal disease status, which could improve the prognosis of oral cancer among Sri Lankan patients. And but unfortunately, no study has been conducted in this regard. So this was the first study from these public health perspectives through to molecular perspectives, which assessed the possible role of these periodontopathogens bacteria with pro-inflammatory attributes, as well as the burden of periodontal disease and the oral hygiene. I would like to add, though her paper is based on this microbiome study, it came as the representative subsample of a major study which was conducted in Sri Lanka, a multi-center study which involved actually 6 provinces over the country, the island, and 9 oral and maxillofacial units, because Sri Lanka has a unique public healthcare delivery model in which oral healthcare, including specialized care like oral and maxillofacial surgery, then restorative care, even orthodontics provided, the services are being provided free of charge at the point of service delivery. So we covered 6 provinces across the country and 9 oral and maxillofacial units across the country. And we had a larger, it was a case control study, 134 cases, OSCC patients, and 134 controls, which included benign oral mucosal diseases like including fibroepithelial polyps. And Manosha used for this bacterium, the microbiome study, a representative subsample from the main study.
Okay, thank you. Thank you very much.
I see. You got all the information which made us to do this sort of a research. Am I correct?
So that was a very interesting background about the disease itself. Could you describe for us a little bit about what you found and what exactly was the dysbiotic microbiome that you found associated with this oral squamous cell carcinoma?
Yes. Do you need to know the definition simple definition of dysbiotic microbiome?
Yes, could you tell us what we mean, what we think of by a dysbiotic microbiome,
Yes.
as well as what you
For
found?
me, dysbiotic means changing the normal composition, that is the species richness and diversity, and functional aspects. Usually, this thing, we have to keep this in mind. These are not pathogens. I mean, just like getting an infection, some Mycobacterium tuberculosis, that bacteria get entered into our bodies. Not like this. These bacteria are normal inhabitants of the oral cavity. But due to various reasons, maybe physiological, immunogenical, immunological factors, with immunogenicity, and other factors like beetle feed chewing, which cause this inflammation in these oral epithelial cells. Due to these factors, in any case, when immunity goes down, they are called pathobionts or opportunistic pathogens. So already they are in this oral cavity. They are normal inhabitant, but our immune system always keeps them under control. In any case, if immunity goes down, they will start because they have virulent factors. So when they get the ideal situation due to various predisposing factors, they will start multiplying without immune elimination by the immune system. So likewise, it's not something to be wondered with the cancer. Inflammation is the main, I think, clinical symptom, one of the main clinical symptoms in carcinogenesis, in cancers in any part of the body. So I am talking about the oral cavity. So inflammation is always associated with the cancers, and the immunity, the immune mechanisms are going on because our body does not wait. When neoplastic changes are going on, many mechanisms, the armor, the fighting between two parties, the neoplastic cells and these immune cells with inflammatory mediators, like a fight between two parties. So these people, they will get the opportunistic pathogens or pathobiont. They get an ideal opportunity. With these things are going on, they can escape from the immune system, and that sort of actions like phagocytosis, various, I don't need to tell about those things, immune mechanisms to knock off these bacteria, so they will multiply. Here, we have found, when talking about dysbiotic, so actually dysbiotic microbiome means difference from the normal situation, compositional, or that means species, maybe in the same healthy condition also, maybe like same genera, but species wise, that very much important. When you are talking about this dysbiotic microbiome, have to find out the relative abundance. So in these disease conditions, we have found species diversity and richness was comparatively lower in OSCC tissues. By this next generation sequencing, we have found 688 bacterial species compared with benign fibroepithelial polyps. We have found 810 species. That means species richness is lower. That means elevation of certain bacterial species and the commensals have been decreased because pathogens, they will try to eliminate these commensals. And here we found, because after that we use this sequencing data to find out risk biomarkers or risk panels. For that, we use, let's say, linear discriminant analysis effect size, that special statistical test. So we have found, yes, Capnocytophaga, Pseudomonas, and Atopobia. These three genera were enriched in the OSCC tissues. That means they were differently abundant in OSCC tissues. When it comes to species, we have found certain species of the genera which I told. When considering, yes, Campylobacter conceus, Privotella salivae, Privotella loischi, Fusobacterium species oral taxon 204, then Fusobacterium nucleatum subspecies polymorpha. So these are the species level, these specific panels or risk markers. And we have actually identified, we have analyzed the microbiome, that is fungal part. That is also very much important because, you know, everybody knows the relation between candidiasis and oral squamous cell carcinoma or immunosuppressive conditions like candidiasis and HIV, like that. So yes, when we are coming to this fungal species, Candida albicans, Candida escheri, and Hanella luteola, of them Candida albicans is the virulent type of fungi species. The risk biomarker we found in the dysbiotic status. That means this Candida albicans, Capnocytophaga, then Privotella species, Fusobacterium nucleatum species, and yes, we found Pseudomonas also. So other species, they are well known, putative, periodontal pathogens. Yes, that is the point I want to highlight. They are normal inhabitants, but under various conditions, when immunity goes down, they will, the numbers are being elevated. They are not came from the outside. So this is something different when talking about Cox postulate and single pathogen. Like, that's why I want to educate the audience the difference. What is this polymicrobial thing? So actually periodontitis is a polymicrobial disease with polymicrobial etiology. So that's why I'm taking much time here. So all these species we found except Pseudomonas are putative periodontal pathogens. We have found them in these OSCC tissues. They have got internalized. So I'm going to talk about these mechanisms, how they get into the tissues later on. So after that, we use another, sorry.
Yeah,
shall I keep on
we'll
talking?
discuss that. We'll come to that. I was just interested, picking up, it's fascinating. So clearly the microbiome changes in terms of which species are present.
Yes.
But I suppose one big question is what actually drives the process of cancer formation and cancer development, and the various different steps along the pathway to carcinogenesis. So I wondered what your thoughts are. You mentioned that inflammation is often associated with cancer, and I wondered what your thoughts are on what aspects of inflammation might actually drive the cancer process and how the inflammatory bacteriome might be affecting that.
Shall I highlight the points? Yes, here in OSCC tissues, we have found risk panels because we use the statistics to find out these risk panels or biomarkers. So putative periodontal pathogens were found as risk biomarkers for further research in this dysbiotic microbiome. Yes, your question, yes. I will.
So these pathogens you think they trigger the immune response, and so the dysbiosis is about enrichment of organisms that might interact with our immune response, and that may then trigger the cancer.
Actually, I'm going to explain that. That's why I didn't highlight over here the possible mechanisms. Actually, here we have not done any animal experiment to find out their carcinogenic capacities using various epithelial cell lines and cancer cell lines. But we have done this. We have found this genetic potential of them. So it's very much interesting to connect you guys from their virulent factors to inflammation to oral carcinogenesis process.
And
the point?
Yeah, and one question that always arises from that is which comes first? Does the periodontal microbiome change come first and drive the cancer, or do you think that the cancer might actually also be impacting the periodontal microbiome?
Yes, this is very much important. Like, which came first? Hen, no, chick or egg? Then egg or chick? I will explain like that. What is the correct thing? Hen or egg or I don't know. And here, because that's why the very important part, the epidemiological part, we have selected these patients according to stringent inclusion and exclusion criteria. Actually, we made use of stringent exclusion criteria. Then we excluded unsuitable people. Then we checked the inclusion criteria and we included. So we wanted to find out the microbiome associated with well-established risk factors. So here, actually, according to my knowledge, because I have studied as a freelance researcher on these things, actually, because they got the that's why I used that word, ideal environment, due to this immunosuppression. Actually, because these people, they got addicted to various risk habits, betel quid chewing, high level of alcohol consumption, and smoking. So they can introduce these well-known carcinogens. So I think the carcinogenesis process has been started. Activation of these proto-oncogenes to oncogenes, then suppression of tumor suppressor genes, at the same time retardation of gene repair processes, then cell cycle, various signaling mechanisms. It's a very much complicated thing when talking about carcinogenesis. But when the immunity goes down, because overburdened status for the immunity, on one hand, they have to eliminate these neoplastic cells, using various mechanisms, like using various weapons to kill the enemy. Like when there are two parties are on the wall like that. I would like to make that sort of explanation. Then even a small child can understand the detail. What I'm telling. Yes. At the same time, yes. So they got the opportunity as a secondary cause when these things, because actually these patients, that's why we wanted to highlight, when compared with the FEP controls, OSCC cases have high periodontitis status. She can explain that, I suppose. Please explain. Yes. It is very important to come to this point.
Yes.
Yeah.
We have to mention now these our OSCC patients, they were older males. Actually, we had males. We limited this study according to our exclusion criteria. Females were excluded, and because it is #1 cancer among males in Sri Lanka, so the males were included. Then they were older than the FEP controls. So we are well aware, old age with accumulative periodontal disease burden, and they were very high. They were practicing aggressive forms of beetle chewing with or without tobacco, the amount and the frequency, and the smoking, alcohol consumption, all that will add to their periodontal disease burden, because we are well aware the beetle chewers, smokers, they carry a high burden of periodontal disease. So actually, because of this lifestyle related risk habits, their old age, they carried a high burden of periodontal disease. And we have to bear up in mind, even among these FEP controls, they as well had these habits: beetle chewing, even smoking, alcohol consumption, but not into the extent of the OSCC cases, and they were relatively young. So this is a sort of a wonderful situation. We never know whether this periodontitis, whether that was a cause or coincidence or consequence or a complication of this oral cancer status, right? Because already they were practicing this risk habit, beetle chewing, and most importantly, these OSCC cases, they had their oral hygiene practices were very poor. 1/4 of them were not using fluoride toothpaste and toothbrushes. They were using charcoal and fingers to brush their teeth. So that is also we have to bear up in mind, because these oral cancer patients, they belong to poor socioeconomic status, and they had significantly lower levels of education compared to the FEP controls, and their practices of oral hygiene or to the devices they have used to maintain their oral hygiene was far from satisfactory. This is a point to intervene, right? Depending on the levels of education and socioeconomic status, people should be, especially those who are practicing risk habits, should be educated on maintaining their oral hygiene while stimulating them to reduce or to give up their risk habits.
Yeah, thank you. So
Yes,
it's
that
clearly...
is the, that is the point of view of epidemiologists. So this is the point of view of oral microbiologist or molecular oral microbiologist. As far as I am concerned, these microbes, they played the role here. Because actually they have not initiated the carcinogenesis process, because these people are with well-established risk factors, or that means introducing the well-known carcinogens. So they got into these OSCC tissues. They migrate from periodontal pockets due to this win-win situation. So this is a coincidence or consequence of carcinogenesis process. As far as I am concerned, as a oral microbiologist, yes, when thinking about these immunological mechanisms, innate and adaptive, with those things, actually, when considering the tumor immunology, those aspects, this is actually consequence. Because of this carcinogenesis process, when it was going on, these bugs, they got an ideal situation to come to this status, because they are with these virulent factors which are immunogenic, antigenic. At the same time, they can stimulate the inflammogenic. Inflammogenic means they can promote the inflammation. So this is a ideal case of consequence, not the cause.
Yeah, it's difficult to know, isn't it? Because you have these shared risk factors, and then you have potential for the microbiome or the bacteriome to impact the cancer, and you also have the potential for carcinogenesis to impact the bacteriome. So I think I guess we need some longitudinal studies really to unpick these kind of interactions and what's actually driving what in the whole process. But it's certainly a very complex area. I wondered if you could tell us a little bit about the type of samples that you used in your study, because when we think of the oral microbiome, we typically think of bacteria covering the hard and the soft tissues in the mouth. But I understand that in your study, you were actually looking at bacteria that were deeply embedded within tissues in the mouth. So both your OSCC, your cancer samples, and your controls both included bacteria that are embedded within the tissue. So how do you think bacteria actually get to within the deeper layers of the tissue in these kind of conditions?
Yes, there are various mechanisms. I think already the scientists have found 2 mechanisms. That is very much, I think I don't need to tell the details about these adhesins, the fimbriae of these bacteria. You know, these here we have not found the Porphyromonas gingivalis, but Fusobacterium nucleatum and other Fusobacterium species we have found, they have pili. So they made use of these organelles to get attached to the host cells. Then this fimbriae or pili, they will secrete various protein molecules, and the attachment, it's like lock and key mechanism, because they have to find out the ideal compatible receptors on the surface of these cells. So after that, you know, these things are happening with various signaling work. Just like when you are driving on the road, to go to your destination, you have to follow the traffic lights. Likewise, various signals, pathways are going on because they made use of this chemotaxis to get into the cells. So with these, adhesins, then invasins and other chemical compounds, they will introduce a signal which is powerful enough, like giving a commandment to change the cytoskeletal structure of these cells. So I have read this polymerization of actin and differences in microtubules. They change accordingly when these signals come. So like this, like something like phagocytosis, you know, these bacteria, then they will get the opportunity to get into the cell. That means they make use of cytoskeletal structures. These configurations will change and they will be able to get into the cell. So I have read Porphyromonas gingivalis is the most studied organism of these periodontal pathogens. Then Fusobacterium nucleatum, Privotella species, they have exhibited various, but the mechanism is this: either changing the polymerization of actin filaments or changing the microtubule. I think if you go through the anatomy of the organelles of a human cell and the transport ways and means, can get the idea. For the general public, I think this is a bit difficult to understand, but let's say, like walking on the road according to the signals, traffic light signals. So that sort of mechanisms are being used for them to get into the cells.
Yeah, yes. Well, it's interesting, isn't it, that they can not just get through the cells in the top layer, but they actually can potentially penetrate quite deep into the tissue. And as you say, clearly there's evidence that oral microbes can invade epithelial cells, and potentially they could move through between the cells as well into tissues. But it's very interesting that you found lots of cells. So one other thing I thought was very interesting from your study, I noticed that some of the groups of people, there were just small numbers of people who had very high levels of certain organisms in their tissues, and seemed to have really an overgrowth of certain organisms. And whereas other people had a slightly different microbiome. So do you think that there are, within the oral squamous cell carcinoma group as a whole, there might be subgroups within that of people who have a particular microbiome that maybe is associated with a particular type of cancer, and possibly even with a prognosis for how that cancer might develop?
Yes, this is a very good question. You know, the principle of next generation sequencing. Here, the amplification and sequencing is done using 16S ribosomal RNA. This RNA, 16S ribosomal RNA. After the transcription, it will become 16S ribosomal DNA, right? So it's a housekeeping gene which found in all prokaryotes. So this is the very important part in next generation sequencing, which is called as open-ended sequencing. So I would like to brief you when thinking about the advent of molecular techniques, because Kary Mullis had invented the polymerase chain reaction in mid-1980s. So after that, various techniques like different real-time PCR, various PCR technologies have been developed. Then another close-ended techniques like not sequencing up to, they have developed up to microarrays, various techniques, various, types of PCR techniques like reverse transcriptase PCR, which has been used to identify this coronavirus. Then various other type of close-ended techniques have been developed up to microarrays. Even the difference between open-ended and close-ended means, open-ended means we don't need to know the prior knowledge. We don't need to have the prior knowledge of these microorganisms we are going to identify. But in microarrays, we should know. We can identify a panel of microorganisms like maximum, I think like 70, 80 like that. Here, that's why open-ended means we found all 1,000 bacterial species, all, because only this, even any DNA compatible with this housekeeping gene will multiply. So that's why all when you are doing these things, next generation sequencing, you should know about the clinical relevance. At the same time, that is very important, the surface contamination. And here we use, that's why tissues, not the saliva. I have to point out an important fact. Here we have done the surface sterilization using, I told in our preparatory meeting, was normal saline, but I tried my level best to take the internal parts, internal tissues. But the ideal method is 70% alcohol. Then you can destroy all the superficial contaminant, but have to be careful, have to remove the alcohol residues, otherwise it will destroy in subsequent sort of DNA extraction things. It will destroy the extracted DNA also, but that is the ideal method. So here we have found intracellular this biotic microbiome with the risk panels after applying the high-flown statistics to high-dimensional data, these periodontopathogens. So you are correct. The thing is the variation, because there's no consistency when considering the risk panel of these microbes. Various studies have found various type of panels, combinations, various combinations. But of them, almost all the studies have found Fusobacterium nucleatum and Fusobacterium species. So that is important. Certain even Prevotella, Capnocytophaga. Some studies found Porphyromonas gingivalis, that is the most important red complex member. But there are similarities. You can't, that is the intrinsic limitation of next generation sequencing, because here we are sequencing all the organisms which may present in that tissue. And it is really difficult, the surface contaminations, type of samples, and way of DNA extraction, and the bioinformatics tools which have been used for these species identification of taxa. So those type of things can affect the compositional status of microbiome. Here I'm talking about oral cancer, but microbiome studies can be done for any inflammatory disease. The connection is inflammation. That's why me and Professor Nessar, we, in our publications, we have highlighted the most important thing, not the compositional details, not the composition or the composition or species of the meta-community. The functions are the most important. That means metatranscriptomic studies are much better, and metabolomic studies, they are assessing the functions rather doing, but very important, because we know with these will be when we are talking about these in vitro in vivo experiments, because it's great. I'm going to talk about that in future research question. So it's good if we can make use of Capnocytophaga, Prevotella, these periodontopathogens, if we can make use of them to find out their ability to make sort of, that's why causality, whether they can do any inhibition of apoptosis or cell migration, that sort of thing. At least they can prevent normal cell apoptosis, or whether they can increase the uncontrollable cell proliferation, that sort of things, making various oral normal tissue cell lines, and further their responses with the immune system. They are immune responses using various type of cancer cells. This part is utmost important, and we need information on these aspects. So I would like to brief you. Yes, there are, depending on the site, various risk panels, but there's no consistency, because it depends, because inherent limitations in the methodologies, type of samples used, the surface contamination, then the various degenerative primers used, the sequencing methods, then the data analysis method, definitely they will affect the compositional profile of these bacteria. But functionally, that's why I want to highlight inflammatory bacteria. We have found. Shall I keep on talking?
Well, I'm sorry to interrupt. It's interesting, but we are already over time. So maybe Nick, if you're okay, we could just talk about the clinical applications, because that would be, I think, very interesting for us to know. And we will just take one or two questions of the audience still, but just focus now on the clinical application. If you have if you're okay with that, and if you have any questions on this section.
Yeah. So obviously you've told us a lot about what you've found. So it would be really interesting to understand where you think this might be headed in terms of applying it to the clinics.
Yes. At the end of this lecture, I will brief you, because I think these scientists are very much interested on these research areas. Yes, you speak.
Now I'll talk about the clinical implications of these findings of this paper. Now it's of no use if there are we have to transform the findings to clinical practice to improve the prognosis of oral cancer patients, either to prevent or to control, and to improve their prognosis. Now, in very clear, simple terms, now we found whether further studies warranted to identify whether it's a cause, consequence, coincidence, or complication, but definitely there is a dire need for improving the oral hygiene and improving the periodontal status of oral cancer patients, or even OPMD patients with oral potentially malignant disorders, which is for most of the oral cancers, this is the preceding condition. So the standard protocols of management of oral cancer patients should definitely include improvements of oral hygiene and periodontal disease status of all oral cancer patients across the globe. But for that, definitely you need to assess their periodontal status in the first visit, right? You can use simple clinical indicators such as the plaque scores, calculus scores, the bleeding on probing, or the periodontal pocket depths, loss of attachments. For that, now for developing countries, it's all the time it's not possible to take the radiographs. So even with these simple measures, you can assess their levels of oral hygiene status and the periodontal disease status. So definitely this has to be included as a mandatory component in their management protocols. More, as I said to you before, based on their socioeconomic status and their oral hygiene practices, you have to provide tailor-made or the customized advice, or the awareness and the knowledge, and even you have to change their attitudes. As I said to you, 1/4 of the patients here, they were using charcoal and fingers. It doesn't mean that they cannot afford the toothpaste and all, because of these cultural practices and all. So therefore, you have to highlight the importance of maintaining a good oral hygiene. But it's not easy to say than done, because their periodontal health status, oral hygiene status is affected by the disease process and the treatment. We know radiotherapy and then surgery or even chemotherapy. So definitely in their pre-assessment, before the treatment procedures, peri-treatment, as well as post-treatment, and in every follow-up visit, this has to be considered.
Thank you. And as you say, if the evidence builds up from studies like your one that there really is a link between periodontal disease and oral cancer, then maybe that will give people impetus to control periodontal disease and promote their health way before the cancer starts, and we can start early. Rachel, did we have questions from the audience?
Let me see if this there is one a question or two. Yes, there is one here. We'll just do one because we are really over time, but at least it's important to answer this question. So the question is, are probiotics helpful if there is a dysbiotic microbiome involved in oral squamous cell carcinoma?
Yes, it is. Yes, it is, because these probiotics lactobacillus, that they are the beneficial microbiota. Actually, gut health and mucosal immunology are of utmost importance to maintain a good health. So, as far as I'm concerned, very much important, these probiotics, they will influence your immune system in a positive way. They will actually help your immune system to work against these neoplastic cells. That is, there are various mechanisms, and you can find out these things. Like probiotics are very much important to actually important in the sense they can boost your immune system, gut health, and make your immune system to work against neoplastic cells or cancers by eliminating them using various immunosurveillance, then this recognition of various type of antigens, tumor-specific antigens and tumor-associated antigens. Then when tumors try to escape from these surveillances by making immune checkpoints, then these various things can be happened. The various immune activities are going on, That's why I would like to highlight the fact, very much important when talking about this dysbiotic microbiome and these probiotics. Actually, probiotics are also the beneficial microbes. So they will help. They will help means they will boost your immune system to eliminate the pathogenic microorganisms as well as, that is the short answer, pathogenic microorganisms as well as neoplastic cells. And here actually oral carcinoma cells or any neoplastic change, because before the clinical symptoms come, there's a sort of a various, it's very much the tumor microenvironment or the point where these tumorogenic activities have been started, activation of proto-oncogenes and inactivation of tumor suppressor genes. So our immune system tries to, using natural killer cells, cytotoxic cells, then inflammatory mediators. That is also happening according to the identification of specific antigens. And the armor is always trying to eliminate the harmful pathogen or any chemical stimuli or physical stimuli. That is the inflammation. Inflammation means your response, the host response, how immune system working against the harmful stimuli. So inflammation is always related with these cancers or other inflammatory diseases. Nothing new, because this has been observed for the first time in 18th century by virtue of, I think about the history. So the answer is yes, probiotics are utmost importance even in the treatment of oral squamous cell carcinoma, because it will boost your immune system in the natural way.
Yes.
And yes, because even for periodontitis, when you have periodontitis, that is another treatment modality. By probiotics, actually, you can seize the problem and you can boost the immune system either to eliminate the pathogenic microorganisms.
That's right. And that would be the ideal, wouldn't it? But so probiotics could potentially do that. And but of course, when recommending things to patients, we have to be very careful that the evidence is there. I think you're right; they have great potential, and maybe that's the next area where we really need evidence.
Yeah, further.
Thank you. Yeah. No, thank you for your inputs on that. actually we see that there is quite of a, it's fascinating to see all the mechanism and all the interactions, but mainly we understand the importance of the role of oral pathogens on other conditions. I'm sorry, I will need to close this session now, but I would like to thank you very much, both of you, Irosha and Manosha, and also of course Nick, for spending time with us today and for bringing to our audience first-hand expert information on this link between oral cancer and oral and periodontal pathogens. I would also, we are very, very nice of you to have all these details. It was quite insightful on the paper. And I would also like to thank our audience because of their attention, and we hope you get information for your daily practice and take-home messages to convey to your patients around the topic of this oral systemic link. Now our journey of the Sunsta Conversations Pro has come to a close. So thank you again, all of you, for being with us. We hope actually to see you all next year to embark on a new series. So that's why I would like to ask you to stay tuned, to subscribe to our YouTube channel and to our social media platform as well, in order to know what's next. So thank you all very much for your company during this journey, and see you soon then. Thank you, Manosha. Thank you, Irosha. And of course, thank you, Nick, as well.
Thank you.
Thank you, everybody. Bye.
Bye.
Bye.